Content on the topic Waddington's experiment on embryonic induction. The work of Waddington and Waterman, 1933, has shown that it is possible to cultivate young rabbit embryos in vitro, using the normal watchglass technique in a medium composed of chicken embryo extract in either chick or rabbit plasma. Waddington thought that it may be possible to obtain as complete an account of the developmental mechanics of the mammalian embryo as Spemann and his school have provided in the amphibia. Challenges: apart from the laboriousness of the tissue culture technique with its concomitant necessity for absolute sterility, the rabbit embryo, if not every mammalian embryo, is very difficult to handle owing to its transparency, toughness and stickiness. Transparency: the transparency of the blastocyst and embryonic shield in the primitive streak stage makes it difficult to distinguish exactly the various embryonic organs. Toughness: the toughness of the tissue hinders neat cutting and operating. Stickiness: while the stickiness of the cells is probably the most annoying characteristic of all, fragments of tissue intended for a graft may adhere to the operating knives. It is, however, quite possible to separate the endoderm, hypoblast, and ectoderm, epiblast, and to place small fragments of tissue in the pocket so formed between these two layers. As in the chick and duck, such grafts develop, at least in the best cases, although in the rabbit development of the grafts, as of the complete embryo, is not so frequent nor so successful as it is in the birds. Usually, after the graft has been pushed into the cavity between the ectoderm and endoderm, it sticks to the knife point and is partly pulled away from the desired position as the knife is withdrawn. The grafts then usually lie in the trophoblastic area. Such grafts do not usually succeed in producing inductions. Only 2 doubtful cases of induction by rabbit primitive streak have been obtained, and in both of them it is impossible to be certain whether the superfluous neural tissue is induced, that is originally host tissue, or whether it is derived from the graft. Grafts have also been made of chick primitive streak into rabbit blastoderms and vice versa. Rabbit tissues grafted into chick blastoderms show a tendency towards the formation of many pycnotic nuclei, but this phenomenon is also seen in the unoperated rabbit embryos grown in vitro, and is probably due to some general deficiency of the culture conditions rather than to a specific action of the chick blastoderm. They thought that this deficiency might be due to the use of chick embryo extract and chick plasma, but Waddington-Waterman found that little improvement was obtained in the length of life of embryos grown in vitro when rat extract was substituted for chick extract, and recent experiments using mixtures of chick and rabbit plasma have also failed to give any better results. In both the specimens of chick grafts into rabbit embryos described here, the grafted tissues have formed hollow sacs. Hiriwa, Murphy, and Nicholas Rudnick, using the chorioallantoic technique, found no tissue incompatibility between birds and mammals till a considerably later stage of development. Owing to its comparatively slight tendency to stick to the operating knives, chick tissue can fairly easily be grafted into the embryonic shield of the rabbit. In 2 cases, grafts of the chick primitive streak have succeeded in inducing the formation of a neural tube from the overlying rabbit ectoderm. Case 23-174A. The rabbit blastoderm, which was aged 7½ days after copulation, had a long primitive streak. A fragment of chick primitive streak was grafted into the left side of the embryonic shield. It was cultivated on a medium composed of 2 drops of chick embryo extract, 2 drops of rabbit plasma and 4 drops of chick plasma. The culture was fixed on the 2nd day of cultivation. The process of fixing embryos kills the embryo so its cells could be examined. The chick graft has differentiated into notochord and some neural tissue, and a part of it has formed a hollow sac. Over the anterior part of the graft mass a subsidiary neural plate appears in the host ectoderm. Case 33-201. The rabbit embryo had a long primitive streak. The endoderm was torn away from the posterior part of the embryonic shield during the extraction of the egg from the uterus. 2 grafts of chick primitive streak were pushed into pockets between the ectoderm and endoderm, 1 on each side, about opposite the middle of the primitive streak. The graft on the right side is lost somewhere in the abundant mesoderm of the host, or in the thickened trophoblastic area. The other graft can be seen on the left side. It has formed a hollow sac, near which is an empty space with some degenerating cells. The host ectoderm above this is folded downwards, and from one side of this fold a strand of neural tissue has been induced. The results show that the ectoderm of the rabbit at this stage possesses the capacity of reacting to an inducing stimulus by the formation of neural plate, that is competent to form neural plate, and thus make it probable that the embryonic development of the mammals is influenced by factors similar to those which have become familiar in the amphibia and birds. These experiments are also a demonstration of the extraordinary lack of species specificity in embryonic organizers, the suggestion that inducing agents are therefore present in the rabbit, and are localized in the primitive streak is perhaps logically unjustified but may nevertheless very likely prove to be correct. Thank you.